So Ashwin is the class of 1981 chair professor at BITS Pilani, Goa. He received his PhD in 1991 from the School of Electrical Engineering and Computer Science from the University of New South Wales. Ashwin joined the ILP group at the Turing Institute, Scotland, and with Stephen Muggleton, now at Imperial College London, worked on the development and application of inductive logic programming. From 1993 to 2003, Ashwin was at the Oxford University Computing Laboratory as a research fellow and then university lecturer. At Oxford, he was involved in pioneering applications of ILP systems to difficult real-world problems in molecular biology and chemistry. In 2003, he moved to the IBM Research Lab India as research staff member. In 2009, he was awarded a Ramanujan Fellowship by the Department of Science and Technology of the Government of India. In 2010, he took up the post of professor at the newly formed South Asian University and became the founder dean of the Faculty of Mathematics and Computer Science. He was professor of computer science at IIIT Delhi from 2012, and then moved in January 2015 to BITS Pilani, Goa, as the chair professor. So Ashwin is going to talk today, and his title is mildly provocative, you might think. It's called Systems are Predicates, but I think, as Ashwin will tell you, it's just a logical deduction or a derivation. Hope you enjoy listening to Ashwin as much as I do every time I talk to him. So without any further delay, over to you, Ashwin.
Okay, good. Can you hear me, Indrajit? I assume
yes,
that
we can hear you loud and clear, Ashwin.
Okay, good. Well, I'll start pretty much straight off because I have a lot of slides and I've timed this, and if I stop for a little while, I'll run out of time. I'm at BITS Goa, as Indrajit mentioned. But this work is actually joint work with a number of colleagues, not just at BITS, but at University of New South Wales, at Aberystwyth when they were there, also at TCS Research. The title of my talk is actually a tribute to eminent personalities in computer science, as you will see, but the consequences of their positions has some role to play in what I'm going to talk about today. So this talk I'll be focusing on modeling systems. By that I mean I'll tell you what I mean in a moment about modeling systems, but I'm talking really about systems which we know little about, or know some details, but not all of it, but they're capable of producing data, and the models that we construct from such systems are not standalone things. They are some things which are looked at by real people, and people play a role in the construction of these models by providing domain knowledge in some fashion, and to machine learning engine, which then uses the domain knowledge and data to construct or refine existing models. And this kind of participatory loop is actually critical to the sort of things that I'm talking about today. So what do I mean by modeling systems? Well, there are three kinds of reasonably accepted views: a scientific view in which systems are treated as equations and data, or an engineering view in which systems can be thought of as built up of components with transfer functions, for example, and data, and arguably probably the most general view of systems, which is that they're actually programs and data. So I will be treating in this talk models as mirrors, in the sense that they are intended to be reflections of the system to allow us to understand parts of it, if not all of it, and if not completely, then somewhat approximately at least. So understandability of things in the system is an important aspect of what I will be talking about today. So
Ashwin, sorry to interrupt you. Is it
yeah,
possible to lower your camera just a little bit? We are just yeah, that's perfect, Ashwin. Thanks a lot.
okay. So there are three big ideas here that play a role in my talk. So one of them is that systems are essentially programs due to Gordon Plotkin, and Tony Hoare's long-held position that programs are predicates. The consequences of these two positions. So I'm not going to explain further each of these two positions. I mean, they are entire talks in themselves. But for us, the interesting thing is that that predicates can be learned in from data, and many people have contributed to that idea from the 1970s to the present, and most prominently, probably in recent times, the groups at Imperial College with Stephen Muggleton and in Belgium with Luc Derat, but there have been many others. In fact, Gordon Plotkin was one of the founders of that idea as well. So I will be also talking about system identification in biology. And why biology? Well, biology is extraordinarily interesting. Now, I'm going to show you a sequence of things from a rather old film, but nevertheless remarkable. I'd imagine that many in the audience may not have even been aware of it, but it was made by IBM in 1970s. So it sort of steps through powers of 10 to give you some idea of the range of scales in biology. So you can go from level of the organism to the level of organ, to the level of the cell, to going inside the cell, to going at looking at molecules inside the cell. So you're ranging almost 8 to 9 orders of magnitude. To give you some idea of what that really means, it may be difficult to comprehend that. So it may be easier to comprehend the reverse direction. This is also from the same film. That if you went, in fact, in reverse and stepped outside, you would be going from the level of the person to somewhat of a plane landing to cities, cityscapes on the planet, to the planet itself. And by the time you get to the same scale as what we're talking about in biology, you're sort of a third of the way to the moon as such. At 10 to the power 8 meters. And this is, as I said, is from Powers of 10 from by IBM made in 1977. I hope they won't mind an old IBM borrowing some of their work. So biology is also quite hard because first principles modeling is difficult. It's not quite like physics. It's not that easy to get equations. Now, data instances are also quite few and noisy, and can be at vastly different scales. The other thing is that we want qualitative and quantitative descriptions for the models. So, what is actually needed? So, I'd recommend a paper called "Can a Biologist Fix a Radio?" From about, I don't remember now, 2008 or 2009, I can't remember, somewhere around that time, which came up with three basic requirements. One is that it needs a formal language that can describe complex systems at different scales, and that the language should be convertible into a form that is understandable by experimental scientists, in this case biologists, and that the language should be usable in the same manner as engineers trying to find models. So, has the same properties of having a kind of calculus to be able to deal with it. Which brings me nicely on to what I really want to talk about, which is inductive logic programming. Now, to understand inductive logic programming, the easiest way, I'm not going to tell you how inductive logic programming works. That's going to take too long. I'm just going to tell you what it does. So the easiest way is to think of deductive logic programming. Now, here are two boxes. In the top box, you have a sort of general rule about grandparents and some specific statements about a family. And you can derive or deduce facts from that about the specific details of that family, that Henry is the grandfather of John and Henry is the grandfather of Robert. So now this is all pretty straightforward using rules of inference. There's nothing especially unusual here. It's deductive inference, where it just follows the bottom box, just follows from things in the top. Now, inductive logic programming sort of goes somewhat in reverse. Given that Henry is the grandfather of John and Robert, and some background information about Henry and Robert, perhaps, you're trying to find a more general hypothesis about grandfathers. In this case, that any X is the grandfather of Y if X is the father of some Z who is the parent of Y. Now, this thing at the bottom doesn't really follow from the top two boxes, and it is a kind of generalization. And in that sense, it's a kind of machine learning in the sense that you're given data, you're given some additional information, and you're constructing hypotheses that that could possibly explain the data. So you could run it in reverse with the bottom box and the middle box, then trying to derive the top box. So how it does it, as I said, is something for a different day. But this is essentially what's going on in a kind of basic inductive logic programming. Why is it interesting? Well, it's interesting because it's not about grandfathers, really, is it? I mean, it's about all sorts of other things. So the examples could be, you know, tens to hundreds of thousands of molecules. The background could be general chemical knowledge, not just a simple sort of database, but general rules of chemistry and problem-specific knowledge. And your hypothesis could be about you know, carcinogenic chemicals having certain structural features and so on, which would be hypothesized automatically. Now, I have to pause here to ask, you know, you're probably wondering, why bother with all this, given all the other talks that you've heard so far? Isn't this all a bit out of date, or even a bit geeky or Greeky, I suppose, if you take the logic part of it into account? And my claim is no, it's not, because domain knowledge is important. Human readability, I claim, is important. Invention of new relations which are not known beforehand in background knowledge is important. And logical reasoning is important. And the other clarification that I want to make is that it's not just about rules, but it's about learning definitions of all kinds of things. Rules is one of them, but you could use inductive logic programming to learn constraints, graphs, mathematical models, automata, probabilistic automata, programs. So today we'll be seeing some examples of some of these other kinds of things. So I don't want to focus on rules, but on some of these other kinds of things. So for system identification, I'll be sort of giving you examples of using inductive logic programming in one of two different ways: a kind of single stage system identification, where you go from giving background knowledge. This is part of the feature of ILP, that you give it some domain knowledge, you have some observations, then you come up with some hypothesis for the entire system, or you do it in stages, where you could start with a prior hypothesis. You have observations and background for each stage. It produces a new hypothesis, which is then either corrected by the next learner in the sequence, and it goes on. Now, by corrected, it means either completion or correction to remove some errors in the previous hypothesis. So, given that sort of rather whirlwind sort of start to or introduction to what inductive logic programming does, let me look at systems as equations and data, the simplest kind of thing in that sequence that I showed you earlier. And we'll be looking at models going from ecosystem to organ to extracellular models. So just for the start, I'm going to spend a little bit of time to describe what I call qualitative differential equations. I'm not focusing on the qualitative part because we can only learn qualitative things. No, that's not true. We can learn quantitative differential equations as well with inductive logic programming. It's just easier to talk about here. And for the models that we're looking at, qualitative differential equations are a nice way of looking at the systems that we have. So qualitative statements or qualitative differential equations are qualitative representations of quantitative statements. Things like levels can be represented as positive and increasing. Derivatives have values which are some qualitative value, and so it doesn't have to be just positive and increasing. It can be, discretized into a number of levels. You can have monotonic relationships to summarize entire equations, like, for example, this amount being some level multiplied by level plus 2.3 would be just said that amount is a monotonically increasing function of levels. In fact, you can collapse whole tables into single statements. Of course, there is loss of information, but there's also some benefits. You can, understand things broadly easier, and noise becomes less of an issue once you're dealing with a qualitative representation. So what is needed then to work with qualitative differential equations are axioms for qualitative relations. For example, here are some axioms which will be the ones that I'll be using for most of the models of this kind, just axioms to do with M plus, which says that y is a monotonic function of increasing function of x, M minus, add, minus, multiply, and derivative. So these are relational definitions of x's and y's when x's and y's are represented as qualitative values, positive increasing, positive decreasing, steady, all these sorts of things. How do you define these relations? Well, there's a well-known calculus for dealing with qualitative differential equations, and you can just define these, which will constitute essentially the background knowledge. Just as a quick, simple example, here's the sort of very basic, relation between force and acceleration, which is represented as a qualitative model on the left. Now there are some flights which go over the Goa campus here, so occasionally you hear some drone in the background, which can make life a little difficult. Just as an example of a qualitative model, are these relations on the left, which can be very easily transformed to the graphical representation on the right. And I will be using the graphical representation because that is the one that is preferred for the problems that we are looking at. So just to give you an idea that whenever you see a thing on the right, you know that there was something on the left which was actually learned and then converted to something on the right. So let's start with a simple predator-prey model at an ecosystem level. This is about 10 to the 2, remember, in that powers of 10 film. So here L is an ILP learner which can take observations. What are the observations? Well, there'll be time series data of predator and prey populations. What is the background? Well, it's definitions of these M plus, M minus, that sort of thing. And what do you get as the result? You get essentially a predator-prey model, which for those of you who know these sorts of things in quantitative terms, is basically two differential equation models, sort of the Lotka-Volterra type model, which then is represented as a graph in a graphical form in this right-hand side of the picture. So this is the correct model. There's in qualitative terms, and it captures most of the things that you would want to know about this predator-prey model. That as prey increases, then how predators increase and decrease with corresponding increases and decreases of prey. Now, let me take you to the next kind of use of ILP in the sequence, where before we started with no kind of model at all. So this one started with no prior model. Let's try something else. Let's step in a little bit further into a finer grain of scale at the level of organs, which is substantially more fine grained than at the level of ecosystems. Let's do something else. Let's give this engine a prior hypothesis to work with. This is actually an approximate hypothesis of the human lung. It isn't correct, or it isn't complete. It sort of ignores various aspects of the lung. So it just has some gases going in, some concentrations, and gases coming out, but it doesn't model anything about the arteries and the veins. And it's possible to construct a model which actually refines that to the correct model, given the data that when you're given data of blood flows. That model is then refined to this one on the right. So this is all done automatically. I mean, obviously, I'm not talking about sort of manually correcting it. It's done. The correction is done by an ILP program that takes in this model on the left and produces the model on the right, which has the requisite kind of blood flows and things. I'm not really going to go through these models on the right to say, look, this is what this is. But you can understand, if you understood the F equal to MA model, you understand that there is some kind of differential equations, monotonic functions, all these sorts of things involved, and that hopefully it captures what is understood in biological terms, and it does. Now, let's make this even more complicated and look at the glucose-insulin path, which is now at an even more finer grain of detail. Glucose-insulin, as you know, glucose is the main source of food, main source of energy for us, comes in from food. It gets used directly by the nervous system, but the insulin part of it models its use or regulates its use by muscles, and in conversion into fat by the liver, and so on. Once the glucose is taken up by the muscles and converted into fat, then it falls, and then we have the level of insulin correspondingly falls. So we have a kind of feedback loop set up, which performs the regulation. It's like a sort of thermostat in some sense. So I should acknowledge here that my daughter draws all these drawings for me. So hence the sort of use of pink colors for organs and things like that. So left to me, I would probably not do that, but you have to give artists their license. Anyway, so now we're talking about multi-stage system identification, starting with no particular hypothesis in this case, but an intermediate hypothesis is produced, which is just the modeling of the insulin production part of it. And then that model is then augmented with the glucose production part of it. Now, somebody is going to say, who decided which stage, how to break up these things into which stages. In this particular case, it was decided by manually because that's what we wanted to do. We wanted to model one stage at a time. It is possible to consider automatic decomposition of stages, but that is a hard problem. It's sort of similar to a function decomposition problem, which is hard to solve. But we're not in the business of trying to automate everything. We're trying to understand things in step by step. So it's okay at this point to look at multi-stage identification with stages coming up one after the other. Again, you'll see that here, this time around, the final model is not a correction, but a completion of the previous model. So that gives you some idea of how to build models with equations. As I said, quantitative equations are possible, but the qualitative ones are more interesting in some sense because it allows you to see them as these graphical forms. But now let me move on to the next stage in that sequence I said earlier of looking at systems as components and data. And we'll be looking at an even finer grain of biological detail, which is an intracellular pathways. So up to this point, we were outside cells. Now we'll go inside cells. So the example that I want to take is the glycolysis pathway, which follows quite naturally from the glucose-insulin regulation, because glycolysis is how glucose is converted into energy. You're not supposed to read all these 10 equations, but it takes 10 reactions to convert glucose into energy, which is at the bottom, which is those ATP guys who appear on the right-hand side. The things in brackets are enzymes which speed up these reactions. Now, the difficulty here is that if we use the qualitative representation of differential equations, then you need about 100 relations to identify glycolysis from data. This can be done, but it's hard. 100 model with 100 relations. So far, the ones which I showed you had about 10 to 12 relations. So, so it's probably easier to think of things at the level of a slightly higher abstraction. So the two things principally involved in these reactions are metabolites and enzymes. Now, each one of these does have a differential equation representation, and this is it. So there is a corresponding qualitative representation of a metabolite. Similarly, there is a differential equation representation of enzymes, and there is a corresponding qualitative representation of enzymes. So, but you could sort of treat these as sort of black boxes and sort of simple circuits, if you wish, that you wish to consider in blocks. And instead, think of modeling things at the level of qualitative reactions. So what is a qualitative reaction? Well, qualitative reaction is something that moves a state, moves things from one qualitative state to another qualitative state. Now, here are all the metabolites involved in glycolysis. On the left-hand side, you see the qualitative states before one or more reactions, and on the right, there are some the same metabolites after one or more reactions. So we don't know how many reactions, but let's say some number of reactions, maybe just one. So you can write some additional domain knowledge, which basically tells you what it means for a qualitative reaction to have taken place, which basically moves a state from state S1 to state S2, using some reactants and producing some products. And the qualitative reaction will essentially decrease the level of the reactants and increase the level of the products. Now, decrease and increase are essentially things that you can write in terms of adds and minuses and pluses and so on, and using the same qualitative calculus that I was telling you before. So rather than going down to the level of the actual differential equations, you can stay at a slightly higher level. And you can, once you do that, you're able to then consider learning glycolysis much more efficiently. So the data going in are pairs of qualitative states, for example, and the background is there are definitions of qualitative reactions, and the result is now again a graph, this time a kind of bipartite graph, because these qualitative reactions are very nicely represented as bipartite graphs in which the enzymes being used as reactants come in to one of the, there's an arrow, inward arrow to the enzymes from the reactants, outward arrows from the products. So it's possible to build, convert the output into a bipartite graph very easily. Now you can add all kinds of domain knowledge to this. So this part of it is just basically saying that a qualitative reaction just increases the products and decreases the reactants. But then you can add all kinds of additional domain knowledge, which is concerns the use of about feasible reactions. So mass balance constraints, for example. We also know things like, oxygen cannot be converted into hydrogen and oxygen. So we know this. So we can state that as part of the kind of mass balance type reactions. There are also bond type energetics, which we know that it's not really possible to have reactions that break, say, more than three bonds. All of these will end up constraining the qualitative reaction and therefore constraining the number of edges in this bipartite graph. So, and that makes the system identification task much more efficient and substantially more. Sort of when we include these kinds of constraints into the system identification, into the background knowledge, you can speed things up by almost 2 or 3 orders of magnitude. So even more background knowledge, for example, would be to treat a sequence of pathway components. So rather than just treating things at the levels of metabolites and enzymes, you can sort of think of it as a kind of integrated chip now, with a generic pathway chip, which will be configured as we learn the system. So kind of like an FPGA, if you wish, for biological systems. So a pathway now is just a sequence of qualitative reactions, where qualitative reaction was the increase and decrease as before. So you can have, n such qualitative reactions hardwired into one of these things, and maybe, I mean, of course, this is just the general definition of a pathway. When you're doing the system identification, you have to decide, the program has to work out which of the reactions have to be hardwired in, and you can do glycolysis again, this time using pathways as background knowledge, and giving data for multiple stages, we are able to identify multiple pathway blocks that together, so given 3 stages, it identifies 3 pathway blocks, which are the correct pathway blocks for the 3 stages of glycolysis. You remember a long time ago, I showed you those equations for glycolysis. So these ones, so they're in 3 stages, really. That's what those horizontal arrows were about. So there are actually 3 stages of glycolysis, which correspond to 3 sets of equations, which constitute specific pathways in this final model. So the use of pathways and qualitative reactions brings me quite naturally to the final kind of system identification that I'm going to show you, which is about programs, treating systems as programs and data. And now we're going to be further into the cell, into signaling, and into the nucleus. So we're talking about signaling and regulatory pathways. Now, pathways and Q reactions are essentially computations. If you think about them, what they're doing are some kind of computation, moving a state from one state to another. So you can think of this as a kind of biological computer, which is working away. Now, in the 1980s, Gordon Plotkin came up with this notion of structural operational semantics for programs, arbitrary programs, which consisted of three different parts. One was a set of configurations, which are roughly equivalent to states, a transition relation, which tells you how to go from one configuration or one state to another configuration, and a meta interpreter that traces the computation by a transition system. So a transition system, and this is the only real logic or formulae that I'm going to show you in this talk. So a transition system's computation can be understood by the definition on the left over here. So let's look at the second thing, which is the easier one to start off with. So if you want to go from a state SI to state SF with a transition system, you pick a transition which moves you to some point, and then you go from that state from SI to S, and from then you do a computation from state S to SF. Obviously, if you're at S and your final state is S, there's no computation to be done. You just stop. That's the base case. So a meta interpreter basically looks at sequences of states and transitions involved in those sequences to work out what the computation is. So what is a transition? A transition basically is something that has some preconditions and some postconditions, and it moves something from an input state to an output state. So that's all. Now, this is the general setting for Plotkin's structural operational semantics, but you can see already that something like a pathway or a qualitative reaction is very much like this. But what this allows us to do is to treat things much more generally. We can now have all kinds of domain knowledge included in preconditions and postconditions, and the definition of a successor relation. The interesting thing about the operational semantics of programs is defining different transition gives you entirely different automata. So one kind of transition definition will give you, say, finite state machines. Another one will give you a Turing machine. Another third one will give you, say, I don't know, probabilistic automata, that sort of thing. So it's a very, very general framework for defining computation. And it also has the hooks for us to be able to write domain knowledge in a very general purpose way. So, so things like, you know, mass balance, constraints, minimum program concentration levels, all of these things can be included in this. So system identification now becomes learning these transition definitions. So you have a general transition system, which is the one on the left. You have to learn the definitions on the right. So the system identification task becomes learning these definitions of these transitions. Now, there's a little bit more, and this is the first time something else is going to make an appearance in this talk, which is not only do you have to learn the transition definitions, but you're going to have to find the most likely sequence of transitions that resulted in the data that you've seen. So our ILP engine now takes prior hypothesis as before, which is now prior transitions. The data are the data for the stage. The background knowledge is the generic transition system that I told you, meta interpreter, which is needed for the interpretation of that transition system, and specific domain knowledge that you may have for this stage. And the output is a set of transitions, but actually it's a probabilistic selection of the most likely sequence of transitions that resulted in the data that you're seeing. So for the first time, I'm now introducing the notion of probability, which I hadn't so far referred to, but actually in inductive logic programming, probabilistic selection and probabilistic modeling plays, probabilistic calculus plays a substantial role in actually selecting hypotheses. So I'm going to give you an example of using this kind of ILP engine to identify a specific kind of automata called Petri Nets. So that I told you that the transition system approach allows us to define any kind of, practically any kind of automata. We've chosen to do it as Petri Nets because Petri Nets have a long history in biology of representing systems. So I'm now only going to show you sort of pictures. So, you know, an ILP engine with observations, background knowledge of transition systems, meta interpreters, so on. And this is the kind of output that you would get from such an engine. So this is a Petri Net. A Petri Net is also a bipartite graph for those of you who are not aware of it. But it's, you know, there are some circles and there are some rectangles in this graphical representation, and it's a bipartite graph. So you get this kind of bipartite graph, which allows you to use standard Petri Net sort of things to simulate dynamics and so on. Here's another example of learning what is called the MAPK cascade, which is a way of transferring signals from the cell's surface, for example, to the cell's nucleus, in which for Petri Net fans, I suppose, if there are such people, this has things like redarcs, as you will see, which are these arrow edges with arrows on both sides. It's a kind of model in which things produced in one step are then used to trigger off things in the next step and so on. It's a kind of cascade type model, again, learned as a transition system. And now the third kind of example, which is to use an existing set of transitions. These guys in dark black boxes were existing transitions. One of them is actually the MAPK cascade from the previous one. And then these are then used to learn a much more complicated model of transferring signals in yeast during the mating cycle of yeast down to the nucleus. So these are examples of fairly complex transition systems, all learned using an ILP approach. So the time that I have left, which is how much, Indrajit?
Maybe we can go on for another 5 to 10 minutes,
Yeah.
Ashwin.
Yeah, okay, good. So in the time that I have left, I'll return to this question of why bother. And the reason actually that we bother with this kind of machine learning is because we're actually interested in constructing in not so much AI, but in IA as such, intelligent assistance. So what you've seen so far is a sort of example of models constructed specifically with the idea of a human in the loop as such. So, you know, there is a biologist who gets observations from some biological system and provides domain knowledge to a modeling system, which then produces models and some kind of understandable explanation. And there may be further round of experimentation. in fact, now there are modeling systems that can do the experimentation as well while it constructs the models. So there are, when building IAs of this kind or intelligent assistants of this kind, there have been two principles that that have basically motivated the work that I've described so far. One is that we have to be able to tell machines what we know in a sufficiently precise way. And two, that machines have to be able to tell us what they have found in a sufficiently understandable way. And I'm fully conscious that it does not follow that machines have to represent or process information in symbolic logic to satisfy these principles. Certainly, they don't have to do it as logic programs, as most ILP does, but that's not the point. The point is that these principles are important, and if you can find better ways of doing it, then I would be the first one to adopt such a mechanism, I think. So the actual so ILP is an umbrella term which you should not focus on the specifics of the implementation of it. So one of the things is that consequences of the kinds of problems that we have been looking at for some time is that black boxes are not an option. So when they're not an option, what are options? Well, a white box is an option where you can take in what you know and come out with some predictions and what has been found. That is roughly the direct the way that I've described today. But that's not the only way to do it. You could use a gray box, which could have some black box and a white box combination, and this sort of thing is done, can be done, and is being done, where actually the background and observations go into this combination, and predictions and explanations come out. How do they do it? I mean, each one does it in a different way. There are hierarchical ways of doing it. There are sort of joint ways of training black and white boxes together. But this is all a talk for a different day. I mean, I could talk entirely on just this slide for the whole hour. So, in summary, what I hope you've been able to see is our own journey of powers of 10, going from ecosystem down to the nucleus, but identifying along the way systems that started using simple mathematical relations and ended up with probabilistic automata. And that has been possible because of this this insight by these two very well-known people of systems being programs and programs being predicates, from which it naturally follows that systems are predicates. But that was the title of the talk, but the content of the talk is really on this footnote that programs can be learned, predicates can be learned, and ILP is one possible way of doing that. So with that, I'll stop. If you want to know more about ILP, there are many groups working on ILP around the world. I follow these ones along with some others. I'm sure you'll find many others doing many interesting things. There are some papers relevant to the material that I presented in this talk, which you can look up on the web or somewhere. Happy to take questions.
Thanks a lot, Ashwin, for the excellent talk. I hope I'm audible. So again, please, if you have any questions, please post them under QA. Here's a question from Gautam, a very specific one. Can our higher level predicates, enzymes, abduced automatically? If so, how?
Yeah, so the invention of new predicates is So there are various things which happen, which I have to distinguish at this point. One is the invention of a, so it's easier to talk about it in database terms. So invention of a new relation is like inventing a whole new a whole new table, for example. That can be done, but it requires some degree of higher level specification on what kinds of relations should be invented. Otherwise, you'd end up a kind of bias-free invention can end up being very unconstrained. Invention of columns in a table, which essentially are like variables in a relation, is very easy. That happens automatically. So given a table, if you're missing some columns, that can be done very easily. When you're talking about inventing entire relations, that can be done, but it is harder. How will require you to look at things like current ILP work on learning using higher order definitions. So inventing a relation will mean that you'll have to be able to talk about relations as variables that you can deal with. So that requires higher order learning. So there is work which has been done on that.
Thanks, Ashwin. There's another question from Shwarya. This was posted before you mentioned probabilities. So his question is there a notion of confidence that can be associated with hypothesis? For example, a hypothesis obtained from fewer volumes of your examples, data examples, or vague, imprecise domain knowledge. How do you associate confidence?
Yeah. So as I said, I deliberately have not gone into the probabilistic logic part of it, but there are several ways in which probabilities are used in inductive logic programming. The simplest way is for hypothesis selection in the usual kind of likelihood prior sense adopted by Bayes. But more interesting, more complicated ways are to have probabilities associated with the predicates itself in domain knowledge. So if you knew something about the domain knowledge, but you were not very sure how reliable this was, you could have statements of belief associated with that. In that case, then the whole calculus changes from having to deal with just relations to having to deal with probabilistic relations. And that is also been looked at. And so the result then would then be not just the fact that that you're using relations, but you are using relations with probability. So the final result will also be a relation with some probability, which then you can decide whether you believe that or not. So you can then start doing all the usual sorts of things that you can do with probabilistic formulae. So you can then start, making your final prediction as a weighted sort of vote across all these different relations that you have learned. I've deliberately picked system identification because it's a very specific kind of activity. You want to understand a particular system. It's not, it's much more preferable to understand the system as saying that I think that these are the components and this is how they're connected, rather than saying that these may be the components. They may also be all these other kinds of models, and for the system activity thing, probability is easier thought of in the selection step, but there are, of course, ways of doing it with much more fine-grained levels. Yeah.
Thanks, Ashwin. I had another question. I don't know if there's a short answer to it. So you talked about programs, systems, computation, predicates, and I'm wondering where neural architectures and neural computation fits into this picture. And there's a reason why I'm thinking about this today, because I'm still trying to digest Professor Hinton's talk from today morning. You're probably there for part of it. So he drew a very interesting distinction between layers and levels in a neural architecture. And his interpretation of layers, it seemed to me to be more like steps in an iterative computation. So in that context, do you think there's a connection between what you're talking about as systems and programs and computation and neural architectures?
So let me answer this in two different ways. One is that how do I see neural architectures fitting into the kind of things that I talked about just now? And there I have a very clear and useful way that we have found uh, for neural architectures, which is in a hierarchical way where neural architectures are used as low-level extractors of relations, including the kind of taxonomical kind of relations that may be that may exist in the data, and then using those relations uh, by a symbolic learning engine to construct models further. So uh, this is not a surprising way of doing doing uh, the sort of learning process, because um, there are many problems for which uh, neural techniques are very good at extracting things like simple relations from which you can build up much more complicated things. And the sort of results that we're getting there are pretty good. I mean, they show that um, things can work very well. The second way in which I have been looking at combining neural and symbolic learning is actually in reverse, where the sort of symbolic information, which is the kind of domain information that I talked about through this talk, is actually provided to a uh, neural network or a neural architecture. And there is no doubt that providing domain knowledge, if you can do it well or sufficiently precisely to a neural architecture, makes a huge difference to their performance. So we've seen enormous jumps in performance when existing domain knowledge is provided to standard neural techniques. Um, Now that domain knowledge might be of the background knowledge type that were used by these ILP engines, or it can be the background knowledge along with sort of interesting new relations found by ILP engines. And just providing the domain knowledge gives a big jump. Providing sort of symbolic relations learned by by an ILP engine provides an even bigger jump. So those are two ways, immediate ways, practical ways that I know that we've tried and tested and they work. Now, the other aspects of things which I don't know much about, but I think are entirely feasible, is what I was alluded to at the very end of my slide, that I don't think that logic programs or are they necessarily the representation language. So I can imagine there will be neural architectures that will do uh, or something close to logical reasoning, um, and they can be used as mechanisms for uh, learning logical statements, um, and then those logical statements could then be obviously much would be much more understandable as such to people. So I don't see that as at all being you know impossible. I think that is very possible. So at the moment, this is the machinery that that I've been using, but I'd be very happy to use something else which is capable of systematic logical reasoning, whether it's a neural engine or a DNA computer. I don't particularly care that much. So
Thanks a lot, Ashwin. We would love to go on. Unfortunately, there's another session starting in the same room at 2 o'clock. So we'll have to stop here. So again, thanks a lot on behalf of the organizers for agreeing to give your keynote talk. We thoroughly enjoyed it. Unfortunately, it'll have to be a virtual round of applause from all of us. We wished we could have had you in person. Maybe that's for another time. Thank you so much, Ashwin.
Oh, thank you.